You're listening to Scientific Giants Who Changed the World. Each episode follows one person far enough into their work that the world they changed becomes visible. Today, we follow memory itself, not as a metaphor, not as a feeling, but as a biological process. We follow it into synapses, into molecules, into a small sea creature that barely seems capable of remembering anything at all. And we follow the scientist who insisted, against deep skepticism, that even the most intimate aspects of the mind must leave physical traces. His name is Erich Kandel. Erich Kandel was born in Vienna in 1929, into a Jewish family, at a time when Europe was sliding toward catastrophe. His childhood ended abruptly in 1938, when Nazi annexation turned Vienna into a place of terror for Jewish families almost overnight. Kandel's family fled. They escaped to the United States, carrying little with them except memory itself, displacement, fear, and the experience of a mind shaped by trauma. This personal history mattered. Kandel would later say that questions about memory, identity, and how experience changes the brain were not abstract curiosities for him. They were lived questions. He trained first as a psychiatrist, drawn by Freud, psychoanalysis, and the idea that the mind could be understood through meaning and narrative. But something unsettled him. Psychoanalysis spoke eloquently about memory, repression, and identity, yet it offered no mechanism. It described effects without explaining how they occurred. Kandel wanted more than interpretation. He wanted explanation. He wanted to know how experience becomes biology. So he made a radical turn. Instead of starting with complex human minds, he moved in the opposite direction, toward simplicity. If memory is a fundamental biological process, he reasoned, then it should exist in some form, even in very simple nervous systems. And if it exists there, perhaps it could be understood at the level of individual cells and connections. This decision ran against the dominant culture of neuroscience at the time. Many believed that higher mental functions like memory could never be reduced to cellular mechanisms without losing their essence. Kandel rejected that divide. He believed that understanding mechanism would not destroy meaning, but ground it. To do this, he chose an unlikely experimental subject, a sea slug called Aplysia californica. Aplysia is not elegant. It does not think. It does not solve problems. But it has a nervous system that is simple, accessible, and unusually large at the cellular level. Some of its neurons are so big you can see them without a microscope. Importantly, its reflexes are easy to study and can be modified by experience. In other words, it can learn. Kandel focused on a basic behavior called the gill withdrawal reflex. Aplysia retracts its gill as a defensive response. This reflex can be modified. If you touch the animal repeatedly without harm, the response weakens. This is habituation, a basic form of learning. If you pair the touch with something unpleasant, like a mild shock, the response strengthens. This is sensitization. These are not metaphors. They are observable, repeatable changes in behavior. The question Kandel asked was simple and revolutionary. Where is that change stored? By tracing the neural circuitry involved in the reflex, Kandel and his collaborators were able to identify specific sensory neurons and motor neurons involved in the response. Then they examined what changed when learning occurred. What they found was profound. Learning did not require new neurons. It did not require rewiring the entire system. It involved changes in the strength of connections between existing neurons, synapses. In short, experience altered synaptic transmission. When habituation occurred, synaptic connections weakened. When sensitization occurred, they strengthened. These changes were mediated by chemical signals, changes in neurotransmitter release, and later by changes in gene expression and protein synthesis. Memory, at its most basic level, was not an abstract imprint. It was a physical modification. This was a turning point in neuroscience. Kandel demonstrated that learning produces measurable, biological changes at synapses. Memory had a molecular signature. He went further. Kandel showed that short-term memory and long-term memory are biologically distinct. Short-term memory involved temporary changes in synaptic strength, often through chemical modification of existing proteins. Long-term memory required new protein synthesis and structural changes at synapses. In other words, remembering for minutes and remembering for years are not just different in degree, they are different in kind. This insight reshaped neuroscience. It explained why some memories fade while others endure. It connected memory to gene expression without invoking mysticism. It suggested that learning literally changes the structure of the brain. Kandel's work also bridged levels of explanation. What begins as an experience, a touch, a shock, a lesson, leads to cellular signaling, molecular cascades, gene activation, protein synthesis, and altered synaptic architecture. The mind leaves footprints in matter. Importantly, this did not mean that complex human memory could be reduced to sea slugs. Kandel never claimed that. Instead, he demonstrated that the basic principles of learning are conserved. Evolution builds complexity by layering, not by reinventing fundamentals. The same cellular mechanisms that allow aplesia to learn also operate in mammalian brains, including ours. Over time, Kandel extended his work into mammals, exploring how similar synaptic mechanisms operate in the hippocampus and cortex, regions central to human memory. His ideas became foundational. Today, synaptic plasticity is a core concept in neuroscience. Learning and memory are understood as dynamic processes embedded in neural circuits, not as ghostly phenomena floating above biology. In 2000, Eric Kandel was awarded the Nobel Prize in Physiology or Medicine for his discoveries concerning the molecular mechanisms of memory. But the prize citation understates the cultural shift his work produced. Kandel helped dissolve the false boundary between mind and brain. He showed that subjective experience and objective biology are not rivals, but levels of the same story. There is also something quietly moral in Kandel's work. A refugee who lost his homeland, he devoted his life to understanding how experience shapes who we are. Memory is not just storage, it is identity. It is how trauma persists and how healing becomes possible. By showing that memory can change, and that change leaves traces, Kandel offered a biological foundation for both vulnerability and hope. So how should we remember Eric Kandel? As the scientist who took memory out of metaphor and into mechanism. As the thinker who showed that learning alters synapses, that experience reshapes the brain, and that even the smallest nervous systems obey principles that scale upward to human thought. As someone who insisted that understanding biology does not cheapen meaning, but deepens it. If you want a final image, imagine a sea slug withdrawing its gill. Imagine that simple reflex changing over time. Now imagine that same principle operating in a child learning a language, in a musician refining a passage, in a person remembering a face decades later. The scale changes, the mechanism remains. You have been listening to scientific giants who changed the world. Today we followed Eric Kandel from Vienna to New York, from psychiatry to sea slugs, from memory as mystery to memory as synaptic change. In our next episode, we will step out of the laboratory and into the wild with Jane Goodall, whose years among chimpanzees reshaped how we understand behavior, culture, and the boundary between human and animal minds.